Welcome back to this Great Academy lecture in biology, where we're doing the second part of the enzymes. And this is where we get the enzymes to actually work for us. We have learned so much about enzymes in the 60s, 50s, 60s, and we actually make them work. You only have to look at a biological powder, a detergent, to see them working. And so what we're going to do is we're going to get to see an organism that possesses enzymes, and we're going to make them work for us. And that organism is good old yeast. Now, Saccharomyces yeasts, Carlsbergian is the little yeast that makes beer. And it's also, there's a whole lot of the varieties of yeast, and they all love carbohydrates. So that's why they're sweet loving. Now, so this guy, he's a yeast, and he has inside him lots and lots of different enzymes, but he usually breaks up any kind of carbohydrate you give him, and he does it for his respiration. He needs his glucose. So he's saying here, if you give me starch, I'll use amylase. If you give me sucrose, I will use my sucrase. So he has a whole deck of cards of enzymes that he can use. So the other thing about him is he is really great. He can stay alive in a very steady state if you completely dry him out, and then you give him moisture, and of course he starts to grow and actually do budding and respiration really rapidly. So there's lots of them about as they reproduce themselves rapidly by budding. So yeasts are a really important part of the course. We encounter them several times, but I'm going to talk to you about him right now. So here they all are. Now, they don't have eyes. I just made that look funny. Okay, so what we do is we get a sachet of yeast, which you can buy in the local supermarket for bread making, and you trap them in a gel, like a jelly from the seaweed, like the algae, and it's called sodium alginate gel. Now, I know that the last lesson I said, hydrogen peroxide is one of those chemicals you have to know. Sodium alginate gel and calcium chloride are two names that you must know. Now, sodium alginate gel, which we allow to fall. So you put them into a little beaker and you mix the gel, and it sticks. It becomes more sticky with time. You stir them so that these little yeasts are mixed with water, which wakens them up, it basically hydrates them. And now they're ready and active, but they're stuck in a little jelly cage. So what you now do is you draw them up in a syringe. You can see all the little ones in here stuck in there. You draw them up in a syringe, trying not to get any bubbles. And then you drop them through a beaker. Usually I use graduated cylinders, a long, tall liquid. And you leave a very small distance here and you let them fall down, and you get circular beads of sodium alginate. And inside the bead, you have lots and lots of little yeasts. And you put this in a strainer and you wash any yeast that might be clinging to the outside. So you want them all stuck inside this gel, a little bit like little birds in a cage. You have immobilized them. You've caught them and they're sitting there. So you've trapped them in a gel. So when you hear the word immobilized, use trapped. If they say trapped, you say immobilized. So the yeast containing the enzymes, all the different ones, are now in those little beads. And the next thing you do is, in my case, we just use a sports bottle. You put all the little beads into the bottom of a tap funnel. Okay, so this is a little tap funnel. And you put a drinking straw, that's what's recommended. We have that in the in-service to stop them falling down through the little tap funnel. That moves around and the liquid will fall down through it. So they're all sitting in here. And you get the same mass. If you took 2 grams of yeast here and you made them all into a jelly, you just take 2 grams of free yeast and put them in here with 10 centimeters cubed of water. The values aren't important, provided you realize that the same amount of yeast in those beads are here. That's the important bit. Now, just when you've the free yeast in here with the water and the trapped yeast in here in the beads, The next thing you do is give both of them at exactly the same time a food that they will happily eat. So it's usually table sugar, which is sucrose. And when you actually make up the sucrose, make it up all in one beaker and put them into 2 graduated cylinders so that you have 50 centimeters cubed of sucrose solution in both. So that's another thing that's kept constant. So you pour them into the top of these 2 tap funnels and you start your stopwatch. Now, what have I written here? This is a bioreactor because they're living things and they're in a container. That's all that it is. So you have a tap funnel and this is a bioreactor. This one has just free. Now they're identical in every way except the yeast are not trapped in the gel. That's the same as that with the same amount, cut and paste, except the only difference is the yeast are here in a gel. Okay, so in 1 minute's time, you open the tap and you drop a drop into a little container. It's often a dropping tile where there are little dimples that you put a little sample in each time. So the sucrose solution is added to both tap funnels and you keep it warm because it has to have an optimum temperature for all the little yeasts. So I usually use about 25 degrees, so warm enough water with sugar in it. Okay, samples are taken from each funnel at 1 minute intervals. That's the critical bit. People forget that bit. Now, I'm going to have him tell you what he has. Now, this time I've shown you some of his enzymes. Looks like you've given us sucrose, so I'll use my sucrase enzyme. Okay, so let's see what happens with a sucrase enzyme. Now look, this is not the entire thing, it's what's inside him. Okay, so what'll he do? He'll take the sucrose, remember it's a disaccharide with glucose and fructose, and he will hug it out basically. So here's one. This is not really supposed to look so perfect. It will be sort of smooth, and then you get the induced fit model. Now they're hugging, and that's called an enzyme substrate complex. And then this little enzyme breaks that bond and then releases 2 products, one of them being glucose and the other being fructose. Now you can detect if there's glucose present because, now here's the overview. This is the substrate, these are the enzymes, then you make the enzyme substrate complex, and then you make the products, and then the enzyme's ready to do the job all over again. So that's the overall picture. If you take a drop of the sample every minute and you dip one of these Clini sticks into it and then count 30 seconds and run it along the side of this, you will see this one has a small amount of glucose because Clini sticks will detect whether glucose is present. And of course, at the beginning, it'll just be a yellow color and there won't be any. But now what happens? Take a sample every minute of this. The first minute, no. The second minute, no. The third minute, oh yeah, you'd get this color change being that. In 2 minutes, in 3 minutes, in 4 minutes, it will look like that. You take a new Clini stick sample every time. In here, in the 1 minute, it's already this color. It's like the free yeast were able to get to the glucose really fast. So what do the results say? The results show that glucose is formed in both funnels. Okay, but in the immobilized yeast, the glucose is produced slower, not until about 3 or 4 minutes. The yeast beads remained in the funnel, which is really good, already separated from the product. So the glucose is coming out here with the fructose. These guys aren't stuck beside it, and the product is a clear liquid, colorless and clear. Now, if you have free yeasts, the glucose was produced in the first minute. But when you opened that, out came some of the yeast. And that's why I sort of drew this picture to show you the little yeasts coming out with the product. And there'll be lots of product in there. So the yeasts are mixed in with the products. Therefore, the liquid looked cloudy and separation is necessary. It was a really nice practical. Now, are there other things? Well, yes, and you're supposed to know some applications. So I'm going to just move myself over here to let you see. What I've drawn on a large scale, a bioreactor. And here are the beads. Instead of them being in these little containers, they're in here, well structured. It costs a lot to make them, but it saves so much money. So lactose, some people are allergic to lactose, to the sugar in milk. Lactose, the sugar in milk. And when lactose is sprinkled down through beads that have alginate beads that have immobilised lactase, an enzyme that breaks up lactose, which is a sugar. It will break the lactose into very useful glucose and galactose. Very nice caramel. I like that. You know, the stuff in Rolo, the ice, the chocolates, the sweets. Okay, now over here, penicillin. Some people are allergic to penicillin. That's an antibiotic. And if penicillin is percolated, a bit like in a coffee making process, if it's percolating down through beads, and this time the alginate beads have an enzyme called penicillin acylase. And what they simply do is they rearrange penicillin and they make new antibiotics. Really important thing. So glucose, very calorific. And when glucose is in excess, some people have lots of glucose in their companies that they don't want to use glucose. They can actually percolate it down through alginate beads. This time, the beads have an enzyme called glucose isomerase. So in other words, they vary the shape of the molecule and change it from being glucose to the product being fructose. So there's 3 examples of the use of immobilized enzymes in industry. But more importantly, this container, just like the tap funnel, anything that's alive will actually be, and that'll be a bioreactor. Now, here are 3 words people sometimes get confused with. Biotechnology is a type of technology that uses living things, like the yeast. A few minutes ago, we were making him work, or their enzymes, sometimes just the enzymes on their own, like here, here, and here, to make useful products. Bioprocessing is using the enzymes in controlled reactions to produce products, same sort of thing. And then industrial fermentation. Well, you learned about fermentation when you were doing respiration with Marie-Therese. And when you were doing anaerobic respiration, that's another word for fermentation. You can make alcohol if it was a yeast, like good old yeast, the Saccharomyces yeasts, Carlsbergian, but you could also use the enzymes to make different things. Okay. Fermentation could look very like this. Now, you're not expected to be able to draw it. And all you'd have to do is make sure that everything is just right. So basically, you'd put your ingredients in, the reactants, which would be the substrate in this case, and the enzymes like yeasts and its enzymes. They'd all be put into there, and you'd be giving them the quantities. Now remember, enzymes have to have an optimum temperature, an optimum pH, and everything has to be just right, which is what they will do. And so here is how it's done. Put in the bios or their enzymes, and you mix them with the substrate and get the temperature right, the pH right. The other conditions are maintained, the right kind of hormones, depending on what you want to do. And then you actually collect the product, or you might do it in one process called batch process. It depends. You have to have a sterile environment because microorganisms, other opportunistic ones taking the opportunity, will have, oh, this is perfect for me. I'm going to grow, and they might grow faster, a bit like weeds in a field. So the microorganisms, what do we use them for? We use bacteria to make wonderful yoghurts, antibiotics, and enzymes. We use yeast to make beer and wine, and as a consequence, then we get them to make things like spirits, like brandy, and the fortified wines like port and things like that. And if we stick them, catch them into a dough, they'll produce bubbles of oxygen or carbon dioxide, and that'll raise the food. So all the breads being made with yeasts, the bubbles that, you know, when you slice your bread and the jam goes through, they're bubbles of carbon dioxide, and that's bioprocessing. And then single cell protein, which are these sort of nuts that you give to cattle during the winter when they can't have grass. That's edible protein. That's using yeasts to make them. A bit like Marmite is also made like that. You either love it or hate it. Okay, and then the fungi, bacteria, and yeast are all produce antibiotics. Okay, what's the advantages? Well, you know this reduce, reuse, recycle, which is really, really important. You know, single-use plastic, it's got to stop. It has to stop. I'm now on my party political broadband, but it's desperate. But Enzymes are brilliant. And if you can remember that phrase, reduce, reuse, recycle, which of course you should anyway, then be careful, because enzymes, if you put them into the beads, it's really good that you can recover them. Just like you saw the beads with the yeast in them where the liquid was flowing out, whereas the free yeast in the immobilised enzyme practical, they just flowed out with them, and you'd have to put it through maybe CERT material or something. You'd have to do a lot of filtering and cleaning and all the rest. So reduce the need for filtration, reduce cost. Now look at those two, both of them are reduce. And reused, can I ask you just to mark that it's most of the time you can reuse them, sometimes not, but at least you have them separate from the product. This is my one pager of enzymes, and you can, or our one pager of enzymes. This is where we take everything you're supposed to know about enzymes and pop them on one page. So just quickly, these are the features, the things you have to know. These are the definitions you have to know. These were the experiments that I did in the last lesson. And this is the experiment I did in this lesson. And there's the applications of immobilized enzymes. I don't think I have reduce, reuse, recycle there, but that again is something that you can make. And I often find that if you had your phone and you took an image of that picture from the notes that you're going to get, and you're sitting on a bus and you're trying to remember enzymes, it's all there. And if you look, you can just quickly look back. And most of us are actual visual learners. People don't realize that enough. And looking at that and the image, that's why teachers fill their rooms with posters and they keep distracting you with this picture and this picture. That's where you go to when you're caught in an exam and you're trying to remember. And this is what they are. I call them, or we both call them, Mary Trace and I call them one pagers. And this is the enzyme one. So, thank you for watching this Grade Academy lecture. Until next time, happy learning.